Hello and welcome, dear friends. Let's continue our discussion with methods of gene transfer. In our last lecture, we already discussed about the artificial methods of introducing the gene of interest into a host by using artificial transformation and electroporation. Today, two more techniques to summarize your curriculum, that is liposome fusion technique and transduction. All these methods employ a technique where the gene of interest has to be securely placed inside the host, where the host will then try to replicate and express that gene in the form of either proteins, the protein synthesis like we see in the case of insulin production, or if it is a multicellular system like a plant genetic engineering or animal genetic engineering, then that will multiply into cells and into a whole organism later on. But the whole concept of this gene transfer is to securely place your gene of interest into the host. So first let us see the liposome fusion technique, or the use of lipid molecules to entrap your gene of interest and then introduce into a host. Liposomes, these are fluid filled spherical vesicles which are made up of phospholipid molecules. These are produced from any fatty acid materials like glycolipids, cholesterols, non-toxic surfactants and membranous proteins. In 1960, British hematologist Dr. Alec D. Bangham, he was the first to manufacture and classify the basis of size, composition, charge and speciality of this liposome or the lipid molecules. These liposomes, they work to deliver your gene of interest, and even nowadays they are using it to deliver a drug, means drug delivery, by using this liposome fusion technique rather than a direct cell fusion. So let's try to understand what is this liposome fusion technique. It's a simple method of transformation of your gene of interest, that is this DNA, into a cell. As you can see, it is delivered here by first entrapping it into a lipid molecule. As simple as that. Means if I take some cholesterol or oil and I mix the DNA and just I release a drop of that into water, a ball of that oil will be formed. This is the simplest explanation of how a liposome fusion is formed. But though it is not as simple as I just mentioned, this method, which was first described in 1960s as a model for cellular membranes, is the use of artificial phospholipid vesicles, and this is used for delivery of your gene of interest into the host. Now, using this technique, the DNA is preloaded by two common methods, that is either membrane fusion is done, or the second is endocytosis, where DNA liposome complex is formed. We will see that how that thing ultimately enters into the host by endocytosis. But this complex, it fuses with the cell membrane. Means when you take a lipid molecule entrapped with the DNA and add it into the host cell membrane and the lipid, they fuse, a complex is formed, and once it enters inside, the DNA is released into the host. Animal cells, plant cells, bacterial cells, yeast cells and all these protoplasts, means which do not have a solid cell wall, these are susceptible to liposuction infection, means they should have a cell wall. That is what the fusion is. The fusion is with the cell membrane of the target cells. So this is the technique of liposome fusion. Now if you see here, the hydrophobic head and the hydrophobic tails, these yellow tails, these are hydrophobic in nature, and the lipid heads which are there, these are hydrophilic in nature. Inside there is an aqueous cavity in which your gene of interest, not only gene of interest, proteins, DNA, RNA, anything can be entrapped in this, and this liposome fusion technique is one of the most popular technique which is being in use nowadays. The walls of the liposome, they consist of continuous lipid bilayer organized in the same manner as that of the bilayer of a natural membrane. These membrane can entrap, as I said, proteins, DNA, RNA, anything. Membrane proteins also can be inserted into liposomes. So in the gene delivery technique, we are trying to entrap a gene of interest, that is the DNA. Even drugs are being used. Drugs, DNA, RNA, proteins, they can be linked to this wall of the liposome, which contains it can be highly concentrated at the center, and liposomes, they can protect the entrapped material, in our case the DNA, from phagocytic cells of the immune system when it is introduced in the gene therapy inside the humans. Now liposome fusion technique is usually used for gene therapy also. So most of the references it comes with, it has to overcome the immunity barrier, it has to overcome the degradation when it is introduced in, means the serum degradation, etc. So that is because this liposome fusion technique with the gene delivery is used for gene therapy in humans also. So these liposomes, they can protect from the phagocytic cells of the immune system by the protective layer of the synthetic polymer, which can be used like the polyethylene glycol or the PEG, which is popularly known as. The walls of the liposomes, they contain very specific proteins, hormones or antibodies, means the coating is usually done, and this allows the liposomes selectively to surface, selectively you can say attached to the target cells. Now liposomes, they can be classified either as cationic liposomes or anionic liposomes, means two types are used depending upon the type and nature of the experiment. So this is a phospholipid bilayer, as you can see, before actually entrapping the genes or the proteins to be delivered, this is a hollow structure, and once the genes or the proteins, they are entrapped, we can add it in the lipid and mix it. As you can see, the fusion takes place on the cell membrane, that is the phospholipid bilayer, it fuses to the cell membrane and the gene is delivered inside the cell. If this is, consider this as a human cell, if it is a cancerous cell, then you have a gene which targets the destruction of this cancer cell. So gene therapy, this is a simplest idea of gene therapy. Now what are these cationic and ionic liposomes and how do they function and what is their significance, we can see. When you talk about liposomes, there are two types, two categories of liposomes, cationic and anionic. So first let us see what is a cationic liposome. These are positively charged liposomes which are associated with the negatively charged DNA molecules. DNA, due to the phosphorus, they have a slight acidic, that is the negative charge, and by electrostatic interactions they form a stable complex with these phospholipids. The positively charged liposomes, they associate with the negatively charged DNA and electrostatic bonding is formed. Then neutral liposomes and neutral colipids are used. Here it is very important because when all these things are mixed in a buffer or a buffer solution, the buffer solution imparts a charge on this. So neutral lipids and neutral liposomes or neutral colipids are used as DNA carriers, and helpers of cationic liposomes are used because they are non-toxic in nature and they have a high stability in the serum when they are introduced inside the human body for gene therapy or drug targeting. There are two types of most commonly used lipids, that is DOPE and DOPC. DOPE is dioleolyl phosphatidyl ethylamine and DOPC is dioleolyl phosphatidyl choline. These two are commonly used neutral colipids. These are the examples of cationic liposomes. DNA molecules, this interacts with the positively charged groups of these two compounds, and the overall net positive charge, it allows the complex formation between the negatively charged cell membrane, the DNA, the whole structure which is called as a lipoplex, just like a complex, a lipoplex. This is then followed by the uptake into the cell and then into the nucleus. So this is how a cationic liposome it functions. The liposome mode of action, as you can see, the gene of interest is entrapped into this cationic liposome. So inside the negatively charged DNA, it just enters inside. There is a core concentrate material of the DNA, and this lipoplex is then introduced to the host cell where the phospholipid layer it merges or fuses with the cell membrane and transformation into the host can easily take place. The gene delivery through this molecular design of cationic lipids is very simple. As you can see, the head and the hydrophobic segment, this is a lipid. in the presence of water it forms a structure, a globular circular structure. So the cationic head groups, they are present on the outside as well as in the inside, and in the core there is an aqueous compartment. This when you add water, there is an aqueous compartment which can entrap your hydrophilic DNA molecule, proteins, RNA or drugs. Okay, the hydrophobic part, this can bind to DNA, RNA, proteins or drugs. In this particular lecture you can just focus on entrapment of DNA. As you can see here, DNA and this vesicle, this is called as a vesicle, the round structure molecule of the lipid which is formed when it is added into water, that is when it is mixed with water, is called as a vesicular structure, and when it is mixed with a DNA it forms a lipoplex. Just like it is a word which is inspired from the word complex, just like as it is lipid which is being used, it is a lipoplex. Now the target, it is targeted and entered into the cytoplasm by fusing it with the cell wall. Endocytosis takes place. Now here two things can happen. One is the DNA endosome, DNA will be released by degradation. As the phospholipid is fused with the cell membrane, DNA is released inside, and this DNA enters this nuclear pore. Now this is a eukaryotic system where it is, the DNA is encompassed by a nuclear membrane which contains small nuclear pores. The DNA has to enter into the nuclear pore. So this DNA is released and it enters into the nuclear pore, and inside the nucleus there is transcription and translation, that is mRNA synthesis and protein synthesis takes place, regulation takes place, and the desired proteins will be expressed. Now this happens when the DNA is able to cross the nuclear pore. It happens only if the DNA is not degraded by the enzymes, DNA degrading enzymes. So this has to be overcome. So there are specific helper liposomes which are being added so that those can be degraded and the DNA is released. So this is gene delivery, and this is how the molecular designing of cationic lipids takes place. So DNA, it is entrapped into the vesicle, a lipoplex is formed which enters or through endocytosis, through the cell membrane fusion, it releases its DNA which interacts with the eukaryotic nucleus, nuclear membrane, crosses the nuclear membrane barrier, interacts with the DNA and releases the proteins of your gene of interest. So this is used in gene therapy, drug targeting, etc. The second type of lipid is ionic lipids or anionic liposomes. This is used because one problem which I just showed in the cationic liposome, that is there can be degradation of the DNA by the enzymes lysosomes. These lysosomes, they can just degrade all the complex because some of the DNA molecules, they get entrapped within the aqueous interior of these liposomes. They are not easily released. Okay, so here the mixing is done with, in this case, ionic polymer, which is a modified cationic liposome. Cations, they are modified, and ionic polymer is made to coat with it, and ionic polymer modified lipoplex is formed. Means the basic liposome is cationic, but you have a anionic polymer coated with it, and then you have an anionic polymer modified lipoplex. The divalent cations which are used in this case are calcium, magnesium, manganese and barium ions, which can neutralize the mutual electrostatic repulsion. Now these are pH sensitive due to their loss of stability. They are not stable at low pH. Low pH is in acidic conditions. These are not stable. So these cationic liposomes, they get inactivated due to this acidic conditions, and also they are somewhat unstable when they enter in the body, in the serum. In the human serum, they are not stable. And moreover, these cationic liposomes in some cases have shown to exhibit cytotoxicity. Means inside, when they are injected in the gene therapy, there is some toxic effects on the human body. So due to these factors, and to overcome these factors, the anionic liposomes are used because they have reduced toxicity, They have less interference with the serum proteins, and they are considered as potential delivery vehicles as compared to the cationic liposomes. So these are the two types of liposomes which are being used. Now liposome mediated transformation, this has certain advantages and these have certain disadvantages. Though the disadvantages are very non-significant, the advantages are they are very economic in use, they are very efficient in delivering the nucleic acids into the cells, not only directly into the cells, also they can be cultivated on culture dish when the cell line cultures are used. Delivery of the nucleic acids can be done with minimum of toxicity. Protection of nucleic acids is achieved when it is introduced inside the cell from degradation. Means protection from degradation is achieved very easily, and it is easy to use. It requires very minimal procedural steps, and it adapts to high throughput systems. Means whatever the modifications of the experiments they are recommended or they come across, these lipids they get modified, and your experiment becomes easier and easier throughout. Disadvantages are that it is not applicable to all the cell types. It is restricted to certain cell lines, and it fails for the transfection of some cell lines with lipids. Transfection is introducing the DNA without the three recombination methods, that is transduction, conjugation or transformation, that is a natural one. You use the artificial method of gene transferring into a system, here in this case a lipid, then it is called as transfection. In sometimes it fails for transfection, means when this lipid is introduced into a human cell line, some of the cell lines they are not compatible with lipids. So these are some of the disadvantages of using liposomes. The second method of gene transfer is using viruses, that is transduction. Now having said that, using these viruses, immediately one thing which comes into our mind is that not harmful? Yes, that is the point which is taken care of, and the harmful effect is when the virus multiplies or when the virus replicates. If you cut that part, if you disable the ability of the virus to replicate and multiply, and just use it as a messenger, as a postman who is just going to deliver your gene, just deliver your message as a messenger, then this method of gene transfer is also very interesting and very effective. Let's see how a gene of interest can be delivered into a host using transduction. So transduction mediated gene transfer, it involves the introduction of genes into the host, genes in the sense the gene of interest into the host genome using viruses. Viruses are the prime carriers of your gene of interest. They are used in this methodology because they have certain interesting features, means they are very efficient. First of all, they are the most efficient transferers of the gene from one cell to the another cell, as we have seen in their wild type nature during transduction, that is lytic cycle and lysogenic cycle. There is high level of replication and gene expression of the viral genome system. When viruses are delivering a genome, there is high chance of their gene expression, and even the replication of the gene percentage is very high. The foreign gene which is packed into the virus and which is made into the host cell can also follow these things when a virus is used. Entry of the virus particle containing the gene of interest into the host cell and then into the nuclear genome, that is it can be fused into the nuclear genome. All this is a receptor mediated process. Means if you are using viruses, now the picture which is shown here is of a bacteriophage lambda, which is just used for the what you can say decorative purposes. Don't go on that. We will be using viruses as vectors which are different, like retrovectors, which don't have that tail and all that. They will be entering the human cells through endocytosis. Okay, the entry of a virus particle containing the gene of interest into the cell and then into the nuclear genome of mostly humans, or you can even think about bacteria. If it is being used as a host, then this lambda phage can also be very effective. The vector genome, it undergoes a very complex process of combining with the double-stranded DNA, that is the host DNA, and this depends upon the vector. Now, it should be able to survive the process of endocytosis till the uh, entry of that gene of interest into the host genome. If it is a multicellular system or a eukaryotic system, then again it has to enter through the nuclear pores. We will see that. So how this thing happens. So basically, once again I would like to mention that transduction is a process by which foreign DNA is introduced into the cells, the host cells, by a virus or a viral vector. An example of the viral transfer of the DNA viral genome from one bacteria to another bacteria we have already seen in transduction, that is the natural type of transduction, and this process is called as the horizontal gene transfer. A viral vector is a virus which has been modified in the laboratory environment for the purpose of introducing genetic material into the cell. Means its virulence has to be taken out. To form a viral vector, the genes which are responsible for virulence, that is causing of a disease, is removed, and these genes, now this provides the opportunity to introduce, when the virulence genes are removed, that much space can be occupied by the desired gene of interest. For example, in most of the case insulin production, you can introduce a human insulin gene and use it for gene therapy. Now this process is done in such a way that the genes which allow the virus to introduce into the host genome, that is the gene of interest, should be left intact, and the virulence gene or the replicative viral viral body formation genes should not be formed. So that is the strategy, and the strategy for engineering a virus into a vector. This requires two you can say components. One is the helper virus and another is the vector DNA. As you can see, this is a helper DNA which comes from a helper virus. This is called as psi negative. Psi it is called as psi, and psi positive is the vector DNA. Now this virus which contains the expression cassettes and the viral non-coding genes, these viral non-coding genes are removed, and the essential viral genes, these are allowed to fuse with the vector DNA. Helper virus or the helper DNA, it contains all the viral genes essential for replication. Try to understand the strategy for engineering a virus into a vector. In the final end, we are going to have a vector which is containing your gene of interest. Now this has to multiply, but virulence should not be there. So what is done? First, a helper virus is taken which contains all the viral genes which is needed for replication, but it lacks the sequence for coding of packaging, that is head formation, packaging of the DNA. It is not there in the helper virus. Second is vector DNA. It contains all the non-coding viral elements, means this is the non-coding viral elements, genes. Gene of interest, that is the therapeutic expression cassette, is your gene of interest. If it is used in pharmaceutical therapy, gene therapy, it is called as a therapeutic expression cassette, that is nothing but your gene of interest. And the normal packaging and release of these these genes are also there in the vector DNA, but not in the helper DNA. Now what happens? The viral proteins essential for replication of the vector DNA, listen carefully, the viral proteins which are needed for replication of the vector DNA are produced and then synthesized in multiple copies of the vector DNA because it contains your gene of interest. These structural proteins, these recognize the vector, that is psi positive, but not the helper nucleic acids. As you can see, this viral replication proteins, this is fused with the helper DNA. This helper DNA, it contains all essential genes for replication. So this helper DNA helps in replication of this gene of interest plus the vector DNA, and this results into packaging of the vector genome into viral particles. How this packaging takes place? Because packaging genes are present in the vector DNA. So the strategy for engineering, you are having a packaging genes along with your gene of interest, but virulence is not there, and you are having a helper DNA which contains the essential genes for replication but not packing. So in this fusion technique, you generate a virus which is now ready. This vector is now ready to deliver your gene of interest. Now, this is not one vector. As you can see, many such viruses are formed. So the first part is to prepare multiple viruses which are non-virulent, which will contain your gene of interest, and which will also contain some markers, like the antibiotic markers maybe for testing it on the cell line culture on plates. So this wild type virus is converted into a vector virus which now lacks the virulence. Initially it had virulence, so essential viral genomes are removed and they are only the replication genes are kept. That becomes a helper DNA, and in the second part the packaging genes are kept intact, plus your gene of interest is kept intact, and you have a vector which contains your gene of interest but it lacks the virulence. So for such type of viral vectors, now these are different types of vectors used for especially for, try to understand, using it as a carrier for your gene of interest. Earlier we saw vectors which were somewhat different. Here now you are using it for gene therapy, where your gene of interest will be introduced directly into the host. The host can be a whole human body and his cell lines also. Okay, so for that particular DNA viral vectors, adenoviruses, adeno-associated viruses, herpes virus, lentivirus, retrovirus, these are used. These are used in such a fashion that their virulence is removed and they are used just as carriers of your gene of interest. The principal mechanism of gene transfer by the mechanism of transduction is like this. As I just mentioned, the viral DNA does not contain the viral genes which is needed to make more viruses. It does not contain that. So viral DNA is replaced, that is virulent genes are replaced with your gene of interest, and this virus is introduced into a cell DNA, that is the target cell line. And then when it introduced, target cell is infected with the viral DNA which contains the gene of interest. Here no new viral particles are created, infection does not spread. Now this will be multiplied to infect the host cells. This is the principle, simple principle, and this is primarily used, as I said, in gene therapy. If the pathogenicity of the viruses like adenovirus or retrovirus can be eliminated and the efficiency of gene transfer and expression is retained, virulence is removed, so it does not cause a disease, but its capacity to multiply and infect remains. Infect in the sense not that type of infection which takes place in disease formation, but an infection where only DNA will be released into the target cells. Then this gene is suited for gene therapy. So your gene of interest, it is encapsulated or packed inside a virus. While packaging, virulent genes removed, gene of interest is added. Now these viruses, they are injected just like vaccines, they are injected, and this viruses, it targets the cells. Let us say in the case cancerous cells, and your gene of interest has a messenger RNA and a protein synthesis capacity. Your gene of interest DNA has a capacity of forming a messenger RNA and a protein which can destroy the cancer cells. So this DNA is targeted to all the cancer cells only. It will attach onto the cancerous cells, because you see this genes of the protein or the protein coat of the virus is made in designed in such a fashion that it will recognize the specific cells. In this example, let us say cancer cells, it will attach on the cancer cells, it will release its DNA into the cancer cells, and then that cancerous cells will be destroyed. This is a promising theory of the therapy of the future, and it is in vogue, in use successfully. So let us see example of gene delivery through transduction. As an example, retroviral mediated gene transfer. In this process, transfection is used. What is transfection? Transfection is the process of artificially introducing a nucleic acid, that is either a DNA or a RNA, into a cell utilizing means other than viral infection. Now don't get confused. We are taking a gene, a viral gene, replacing the virulent genes with our gene of interest, then introducing this into the cell body. Now while introducing this into the viral cells, we are not using the method of viral infection, but it is artificial in laboratory, that is called as transfection. So transfection of packaging cell line is the first step. virus collection, means once that is then introduced inside a viral body, you allow the viruses to multiply. Don't again get confused with two terms here. Viral multiplication is taking place because we are keeping the two genes intact, that is helper cells are allowing the replication of the viral genome, and the wild type of viruses whose virulence genes have been removed are still containing the protein formation, that is the maturity and the head formation, the protein coat formation is still intact. So this allows the replication, but there is no virulence. So once that is done by centrifugation, you can collect the viruses and then use this viral culture for injection, that is transduction into target cells. The final part where it is introduced into the target, that is the human maybe, that is transduction takes place there. So let us see these three processes somewhat in bit detail. The first step is transfection of packaging cell line. In this method, the production of replication defective virus can be accomplished. In the first step, means the wild type replication doesn't take place. Wild type which is capable of causing disease is not replicated, instead of which a helper replication gene is allowed. So wild type replication defective virus is generated, then it is packed with transfected, that is packed with viral plasmids. Why we are calling it as plasmids? Because some genes are removed and it is circularized off, it just resembles a plasmid. Without the phage head, it is as good as a plasmid. So it is transfected with plasmids which expresses all the viral proteins which are necessary to proceed with infection. Now here when I say infection, I mean just adsorption and injection of the DNA, no virulence. Okay, so here the viral plasmids, they are delivered into the host cells or they are packed into the viruses and more viruses are generated. As you can see, different types of genes can be introduced and different types of modified viral genomes can be produced. Now here when the plasmids they are introduced, they express all of the viral proteins and they generate infectious particles as well as the nucleic acid sequence of our gene of interest. Means with each viral particle here, your nucleic acid, that is the gene of interest, is also replicated, and each virus, it contains your gene of interest. Retroviral vectors, they produce replication defective or self-inactivating particles. Now these are replication defective in the final end, means when they are introduced inside, they will not replicate, but they will go into the target cells and they will secrete the proteins or act on the cells which are being targeted. So when these are replication defective in the final end, this allows the delivery of the desired sequence without continued viral replication, without virulence. So this process is the first step, that is transfection of packaging cell line. In the second step, you have to actually collect the virus. The first is you pack your gene of interest into a virus, allow the virus to multiply in certain cell lines, and then you have to collect the viruses. The packaging of the cells, it produces viral load or a lot of viruses whose genome you are interested in. The concentration of the virus is collected by centrifuging. As you can see, enveloped viruses and non-enveloped viruses are produced. The viruses are released in the supernatant and the lysate, that is the bottom, it is being removed, the pellets they are removed. So initially viral vectors with the helper plasmids, they are grown on a cell line culture, that is on Petri plates, then they are centrifuged in a specific solution, buffer solution, and the supernatant is collected in the form of viral culture. Viral culture is collected in the form of supernatant and that is used for gene therapy. The purified viruses, they can be stored at -80 degrees centigrade. So the first part, I repeat again, is transfection, packaging of your gene of interest into the viral body. Second is allowing it to replicate in a cell line culture, that is the viral culture collection, and the third is transduction into the target cell. Now this is the part where you are injecting it into the humans, or this is the part where the actual gene therapy takes place. Viral envelope or the glycoprotein, as you can see here, the viral envelope. Now the final product which the virus is containing your gene of interest, it is containing in a viral envelope the glycoprotein, it interacts with the cellular receptor and enters the actively dividing cells. Means this is a human cell, let us say. Now this is an example of gene therapy using adenovirus vector. Now this is the large part, the blue part is the human cell. On the human cell, viral envelope is made to attach. It enters through the cell membrane. The virus is packed in the vesicle, that is the cell membrane. Then as it goes inside, there is an increasing acidity. So the viral spikes, they are just released and they just go on disassembling, that is dismantling of the viral proteinaceous material takes place at the time it reaches the nuclear pore. At the nuclear pore, as you can see, the virus disassembles and delivers the DNA into the nucleus. The viruses will not enter into the nuclear pore. The DNA with your gene of interest is inserted inside the cell, and genetic information from the new gene is transferred inside the molecule, and here the mRNA is synthesized. Now this is the blueprint for protein production. The protein, which will be protein? The protein which you designed in your gene of interest will be produced inside the multicellular system or the eukaryotic system here. So the viral double-stranded DNA of the retrovirus here is not capable of passing through the nuclear pore complex. This requires a breakdown, and during mitosis of the cell, this happens and integration may take place. Actually, the whole virus cannot enter, only the DNA is released inside. The viral genome, it enters inside, goes through the nuclear pore, and the desired gene sequence, here your gene of interest shown in purple color here, is integrated into the host genome through the activity of various enzymes, and the other part of the viral genome is inactivated as it is replication defective. Only your gene of interest is inserted inside. So this process where the virus comes, attaches to the host cell, introduces its DNA, is called as transduction, and transduction is used as a method for gene transfer. As you can say, gene of interest transfer is done by transduction, that is using viral particles. So this is in general the flow sheet or the schematic diagram for what I just mentioned. You can pause this video here and take a look at the whole picture. The DNA which is created by transfection is introduced into the cell line. Either it will remain as it is or it will integrate into the host genome. Two possibilities are there. When it enters into the host genome, a stable expression takes place, and viral proteins, they recognize the Psi plus region, which I just told you, that is the vector genome, and there is a helper genome also. So viral structure proteins, they are formed, that is contributed by the packaging cell line. We have these genes kept, means the envelope, the cell envelope, polymer, proteins, all these are formed and packaging takes place. On the other side, there is replication of the gene of interest by the helper cells also. Finally, the viral proteins, they are packed, and when they are released through these cell line cultures, they are infectious, but they are incompetent non-virulent viruses. These viruses can be collected by centrifugation, and using gene therapy methods can be injected into humans or animals or wherever this technology is applicable. So coming to the end of this discussion, I would like to just summarize the methods of gene transfer. Gene therapy is one of the most prominent applications of this gene transfer technology using molecular cloning or recombinant DNA technology. Gene therapy is used in medicine, and thanks to the advancements in gene editing and the recombinant viral vector development and molecular cloning, I would like to say that the interests of clinicians and pharmaceutical industrialists, they have been regenerated. There is a huge potential market worldwide for this technology, and with the advent or the invention of this technology, more than 12 different gene therapy drugs have been now designed for curing blindness, cancer, immune deficiency, and neuronal disorders. This has come into the research limelight and has become popular amongst the researchers, pharmaceutical scientists, and also recombinant DNA technologists. As you can see here, the methods of gene transfer employed can be DNA viruses, RNA viruses, liposomes, cationic polymers, dendrimers, cell penetrating peptides, etc. So the whole idea is to transfer your gene of interest into a cell and allow it to interact with the host cell and achieve the desired effects. So this is one of the most popular methods and has a huge range of applications. In our next lecture, we will be discussing the screening strategies. Now, in short, we will see once this gene is delivered inside the host, we should have some technology, some method to ensure whether our gene of interest is really functional or not. So, we have to screen to see the efficacy of the transfer method or the molecular cloning process. I will be discussing three methods in short, that is insertional inactivation, immunochemical methods and colony hybridization in one lecture. So till that time, go through this lecture once again. If you have any difficulties, contact me. We will have a good discussion, and also by the time I would like you to go through the references, the links which I have given down below in this video. Have a nice time, do study and gain for knowledge which is higher than what you are today. Thank you. Goodbye.